This presentation on polyqtlR is sponsored by the Tools for Genomics Assisted Breeding and Polyploids Training Workshop and funded by NIFA, the National Institute of Food and Agriculture within the USDA Specialty Crop Research Initiative, and was held on January 15, 2021.
But I would also really encourage you today, if you can, to stick with me as much as possible on this workshop. So to download the files, they're there through the link on the website that you should have got, and it's also probably in the email. So if you can save those, I've saved them myself on the desktop in a folder. I'll just have a quick look at those files since we have a couple of minutes extra. So there's a CSV file there. You can actually open it up as well if you want and just have a look. These are phenotypes that were measured on the rose population we've been talking about in a couple of different environments in Kenya and also in the Netherlands. And I've selected 3 traits that were studied. So prickles on the stem and on the petiole, and also the number of petals. And they were measured across those actually 4 separate environments. So that's the data that we're going to be working with, and we're going to be connecting those phenotypes. That's real trait data, so with missing values and everything. We're providing it as a resource for this Polyploids project. So you can play around with that as you like. But if you can, to load up RStudio and like we did yesterday, open up the workshop file. So it's a .or file within RStudio. If you're first set your working directory there, and then you can click along through these commands with me by clicking Control and Enter. So again, polyqtlR, it's actually, I don't have any slides for today because Chris, I think, has given a very nice and complete introduction to the topic of QTL mapping from diploids through to polyploids. He's mentioned quite a few features actually that are covered in the polyqtl model. polyqtlR package. So I'm just going to dive straight into a sort of a hands-on workshop demonstration. So we install the package and load it, and we also load those data sets. So that's using a phase map list from yesterday. Also the SNP data, so the genotype data that we have, and that was, again, we saved that in yesterday's workshop. So it's the same file, and a phenotype file as well, which is the one that I've just got open at the moment. So I'll probably just close it before Excel tells OR to stop. There we go. So they're the 3 main files we'll be working with. Just to mention, and I think was really nice, Marcelo pointed out yesterday that MapPoly can also deal with files from PolyMapper. We're also trying to get those packages to talk to each other. So if you chose, for instance, to use MapPoly to create your phase linkage map, you will have a MapPoly object. And there's a okay. See, convert. Okay, so there's a function within polyqtlR, which is to convert a MapPoly object to a phase mapless. So if you chose to do your parental phasing using MapPoly, that's fine. You can just convert it to the format that's expected in our package. I suppose I should turn this off as well. I'd be getting all these, excuse me, emails being sent at this time of the evening. Very very dedicated student. So we're getting all these. Yeah, you can use whatever package you like. Of course, polyqtlR was designed with the input from PolyMapper in mind. But what we're trying to do with these 3 days is to show you the workflow all the way from FitPoly through to PolyMapper and polyqtlR. But as I said, you can use different options along the way. And I think that's what really we should be striving for, is that these packages you can switch between and pick the best features of all the packages, so that you don't have to keep figuring out what sort of input file do I need now. So we're going to, I think, at least from our side, try to improve that a little bit further. Okay, so IBD probabilities have been discussed quite a lot. Yesterday they were featured and they were estimated in the workshop by Marcelo. And there was also IBD probabilities estimated using PolyOrigin. So we actually included that as part of the polyqtlR package. We have a function. Originally, it was called PolyOrigin because it took the tetraorigin approach for for phasing the offspring and actually extended it to multiple ploidy levels. But we've now just called it a general function called estimate IBD, and this is the core of the it's a single function. You run it by supplying your marker genotypes, your phase map list. You tell the ploidy level. You choose a prior for your error. Here we're putting a bar of 5% error, and then as before, there's the option to use multiple cores. Now, Chris mentioned, of course, that this is the hidden Markov model that was actually proposed and implemented by Xiaozi in his software. But there is another method, the quick and dirty method, or the heuristic method, which is works for all ploidy levels. And is extremely fast. And that's actually the implementation of that. I must acknowledge Geert van Geest, who's also here today. And he implemented that method in a very nice way. So it's incredibly fast. So if you are working with a species at a very high ploidy level, and these hidden Markov models are starting to go slow, you can just use the very fast implementation that we have here, but you would have to then set method equals, and you would do heuristic. Now, all these things are described in the vignette, but today we're going to be just using the ones, the IBD probabilities as estimated using the hidden Markov model. Now, there was some discussion yesterday also about double reduction. We can also run this function without this bivalent decoding. So this is using the language of tetraargent, but without bivalent decoding. So we can turn that off. And that will give us a set of IBDs which also include the possibility of double reduction. Now I'm going to run it here while I'm talking, and you'll probably notice that in the background it takes a little bit more time to run than the bivalent model. And as you can imagine, if you're allowing also for all these extra genotype classes which have, instead of a single copy of homolog 1, which have 2 copies of homolog 1, you start to add all these extra combinations. And so those numbers that we saw yesterday from Marcelo, the numbers 36 and scaling up to 400, well, they start scaling up much more when you add all these extra double reduction classes. And therefore, your transition matrices start to become much bigger. Now, there's nothing really that complicated about including it, especially when someone like Xiaozi has described it so nicely, and so we have that available also for hexaploids, but at the higher ploid levels, it really starts to run quite slow. And as we said, and as Chris mentioned, we published that paper now a couple of years ago already about looking at what is the impact of including those multivalence and double reduction in your QTL analysis, and it didn't really have a huge impact on the power. Something that I will come back to is the GIC, and it's great that Chris mentioned that I should do that, because well, it's nice to talk about information content, because I think that's much more important than whether you start to model the small percentage of double reduction events. Of course, on the other hand, it is very nice if you have double reduction that you can spot those individuals, especially if there's a double reduction event around a QTL that you're interested in and happens to have a double copy of a homolog, a specific allele that you have information about being useful. But that is more from a more applied or a next stage. But for the general first analysis, for exploratory QTL analysis, it really doesn't add a huge amount to your analysis. Okay. Now, Chris also introduced the topic of interval mapping. Interval mapping can be essentially approximated quite easily with these IBD probabilities, because we have now just in the background there run a hidden Markov model to estimate these IBD probabilities at all our marker positions on this phase linkage map. And remember, there were essentially about 1,000 markers positions on each of our chromosomes in rows. Now that's an awful lot of marker positions, and in the end, the information at those markers is fairly similar to the ones in nearby markers. So we have an oversaturated linkage map when it comes to then the next stage of QTL analysis. You can think of it like in this Markov chain, that the information between neighboring markers is being shared with each other. So those markers are essentially going from single biallelic SNPs to being sort of multi-allelic or multi-SNP, multi-allelic markers, essentially in a local sense. And then, so the information is all kind of joined and shared with each other. So it doesn't really make sense to run a QTL test at every single marker position. But of course, that's what is the output of your hidden Markov model. So instead, we fit splines. So we just simply use the splining functions here that are available in R. Um, and actually, curiously enough, it's actually faster to estimate the IVD probabilities than to fit the splines afterwards. This takes about 40 seconds on my computer, whereas the original function to estimate the IVDs takes about half that time. And that's also because part of this has been, I think, like Marcelo did, has been programmed in C or C++ in order to speed it up. But the splining is essentially just taking those probabilities, fitting lines between them, and then at a grid of positions, here we're doing at a grid of 1 centimorgan, we would then just take the IBD probabilities at those positions. So instead of having 1,000 test positions per chromosome, we would look at something like 80 or 100 along at irregular spaced intervals. So it is a sort of an approximation of an interval mapping approach. Okay, so that is it still running? Oh, what's going on here? Okay, yeah. Well, I'm going to save that, which I think is, well, I don't need to save it because I've probably saved it already. But anyway, so it's always good when you're dealing with data to look at the data. And we'll come back to those IVD probabilities in a while. But first, let's look at our phenotypic data. Now, I must say, probably the polyqtlR package could have had more functions for visualizing phenotypes, but I've just added a few for today, just so that we can look at these traits. And get this out of the way. Okay, you can see what. So I'm just want to, yeah, I'll do that like that. Okay, so these are 3 traits that we're going to be working with. They're much nicer to look at in the vignette. So if you open up the workshop files that you were given, and you go to here, yeah, I spent a bit of time on spacing them out now, so you can see those 3 traits, the prickles on the stem and the petiole, and the number of petals, and just how are they distributed across in the different environments. So in Kenya and also in Wageningen, either in the summer or in the winter time. So the number of petals, for instance, the distribution is quite similar across environments. So it seems to be fairly stably expressed trait, whereas the number of prickles does show more variability between those environments. Of course, usually you can look at distributions of traits by using box plots. It's quite a normal way to examine your data and, for instance, look at potential outliers. So here, for instance, we see some values that are quite high, and I just want to pull out those values. So I want to pull out the phenotypic scores for the number of petals, which are greater than 80, and I want to, and also now missing. So if you look at the output of that, you see that it's actually the same individual each time here. It's individual K50, which had those outlying values in terms of number of petals. So it's probably actually just one of the offspring which had a very large number of petals. You can do that same sort of thing for the other traits. And again, looking at outliers. I'm not going to start removing outlying values, but before you start a QTL analysis, it's always good to look at your data, make some visualizations, make some plots. Sometimes you will see strange points. Again, this is very basic stuff, but it's worth mentioning because a lot of people go straight into an analysis with very fancy models without even looking at their data to begin with. So don't make that mistake. Another way that we might try to look at the data is to compare our traits across environments. So take all pairwise comparisons here between Kenya and Wageningen, and this is Najobo and Nairobi. And again, for the trait, the number of petals, you can see it's quite a tight correlation across those pairs of environments, suggesting, again, as we thought, that the number of petals seems to be very stably expressed, potentially having a more simple genetic architecture than you would have for the other traits, number of prickles, which seem to have a bit more of a cloud structure. But again, those sorts of visualizations can highlight issues and or potentially lead you to consider how you should run the analysis. So that's visualizing the haplotypes, the phenotypes. I now want to visualize the genotypes, and there's a function. Chris already showed the output of it called visualize haplo. And here I'm just taking our IBDs, um, which is a list. I don't really want to go in right now to the full structure. So like polymapper, polyqtlR is just a collection of functions, and a lot of the objects that are useful in the package are lists. So you have to use the $ after it to start finding out what's inside it. So inside IBD, we've got a number of linkage groups, and inside each of those, so it's a nested list, there are all these different things. So the IBD type, the IBD array, that's the really big three-dimensional array with the probabilities in there, the map, the parental phasing, and then some other things that are needed. So it's a little bit like an object-oriented environment, except instead of that, we just use list structures, which is far more simple for me, at least, to get my head around. So that's a visualization of the haplotypes here of the first 9 individuals in the population. So I've just taken out the column names of the dosage matrix. I've skipped the parents, so I've skipped parent 1 and 2, and I started at number 3. So here I've got 9 individuals just randomly. I want to select those offspring, and I want to do a multiplot. So I want to use 3 rows, 3 columns. Now the topic of double reduction, as I said, is bouncing around. So let's have a look at those ones that we estimated using double reduction in our model. And so I just copied the exact same call, but instead now of the IBDs, I put in the IBDs including double reduction that were estimated. And you can see immediately 2 of those individuals have segments that are now colored red. So similar to what Xiaozi showed yesterday, we use the coloring red to denote an extra copy. So these dark blue represents a high probability of inheritance. Lighter blue represents then that there has been possibly a crossover, so there are recombination, so a bit of uncertainty around the positions there. But then this is very clearly evidence of double reduction. So we can toggle back and forth between those and see, for instance, in this individual K03, when double reduction wasn't included, then the model chose to swap around and actually introduce many extra combination events. So on a very local level per individual, it can have an impact on the quality of those IBDs. But as I said, across a whole population, it's a randomly distributed and fairly minor affair. Now, the other thing that we did, of course, is we splined those. So we interpolated those IBD probabilities, and we had an object called IBD splines. So if I just copy and paste the same call and now add the IBD splined, it's just a much smaller, that shouldn't look like that. Okay, it does on this screen. When I was doing this earlier, you could see exactly all these little lines. But anyway, it's just to show you that there is no difference in those IBD profiles. So they look exactly more or less as the original ones, but now instead of having all the positions, they have just a subset of them at regular intervals. So we're not losing any information by doing that interpolation. The next topic is to estimate the GIC. So this is using a function called estimate GIC. And we just, again, provide in this case, our splined probabilities to make it a little bit faster. And what are our GIC? Well, that's this genotypic information content. And I'm going to visualize them just for chromosome 1. And you'll see here underneath these profiles, you've got the distribution of your marker alleles from your linkage map. And then above them, you've got these lines. So these lines represent, relatively speaking, the amount of information you have, or inheritance information you have, about each of those homologs across the whole population. And it's essentially just looking at how close your probabilities, your IBD probabilities, are to zeros and ones. So if you have very clear information, you have very dark lines in your IBD profiles, you'll be very sure about inheritance, and then you have very high information. And that leads to much more accurate QTL analysis than in regions of variable information content. And that was something that we explored quite a lot in this paper. And in fact, through simulation studies, found that there's a very, it's a nice figure, I was quite proud of at the time, a very clear connection between these GIC profiles, which are above, and the position of where the simulated QTLs were eventually detected. So it introduces quite a lot of bias. So variability in this GIC profile actually means that you'll start finding QTLs where they weren't. So this is underneath the position of where they actually were simulated to be, and then the arrows point to where you find them. So you start seeing this effect where all your QTLs start to be found at regions of high GIC. So this variability in the GIC leads to a poor estimation of your QTL position. And whereas in this region here, and this is again work that we did with Christine Hackett over in Scotland, in this region with high information content, you can see those arrows are vertical. So you're finding the QTLs more or less where they should be. So your position accuracy, your precision is very high. Okay, one other point to make, and that's something that I point out in the tutorial here, is that on the one hand, we can use the original marker coding, and that's something that I talked about yesterday, converting the marker dosages. And if you use those converted marker dosages in the visualization, at least, you find a much more meaningful um, connection between, on the one hand, marker distribution, and on the other hand, your information content. So if you start just visualizing your marker alleles using original codings with lots of high numbers, so 3s and 4s, you will look like you've got a saturated linkage map. But in fact, if you use those converted marker scores, it can show you regions where you don't have any information. And you can see, for instance, in parent 1 homolog 4, here we have, from here on, we don't have any coverage of that homolog. And that's reflected in this dropping yellow line, same color, so they correspond, dropping down. So the information content is really dropping off at that end. It picks up a little bit because there is some information carried on the other homologs, and by exclusion, that is picked up by the hidden Markov model. So there is some extra information here, but indeed, it shows you the importance of marker coverage across homologs. And that's something that we very much stress in when we're doing QTL analysis, is to look at this quite clearly using this GIC measure. Okay, so for now, that's a bit of an introduction. We're going to start doing some QTL analysis. Now I'm going to stick to single trait. That's why the topic was called It's a Thorny Issue. We're just going to be looking at prickles, and in particular stem prickles. So the main workhorse of the QTL analysis for this package is a function called QTL Scan. This is using this a model that was introduced again by Christine Hackett, and it's part of the tetraploid SNP map program. So it's I think it's very similar, a weighted regression using the IBD probabilities as weights, but it also allows for blocks. So for instance, here we've got 4 environments. So some level of experimental design is allowed for, and the addition of genetic cofactors. So building up towards a more multi-QTL model, which we'll see in a while. This is quite flexible in. Okay, so we just run our QTL analysis, running this call here, providing the phenotypes. We say the name of the column, of course, the column header for our genotypes, and what is the trait. So that, again, the column header. All this is described in the help menu. Just to say, we don't make any requirements that you have to have the order of your genotypes in the same order as it was in between phenotypes and genotypes, or that you have to have exactly same matching. All of that is handled by the function. The only thing that's required is that the names of your offspring are consistent between your marker array and your or your marker assay and your phenotype. So if there are any differences, then those names are not able to match. So it has to be perfect matching between the names, but the order and whether you've got missing values, all of that is handled by the function. Now, again, a lot of these functions, like Polymapper, they provide output that looks a bit like this. So it's a sort of a markdown type output, and that can be channeled, again, like Polymapper functions, can be channeled to a log file using the log function, a log argument of this function. Okay, so we've run a little QTL analysis. It was just gave us a warning. That's because there are block effects fitted to try to maximize. If there are missing values, they're imputed to an extent across blocks, but I won't go into that right now. And let's just plot the first, there's a couple of plotting functions for QTL plots within the package. The first is just called plot QTL. Probably nicer is to use the plot linear QTL, where it just puts them right next to each other. But you'll see usually this you're associated with defining a QTL as something that comes above a significance line. And again, Chris introduced permutation testing, and that's something that's used all the time, and that's how we set our significance thresholds in this package. But you can do it using this function, this QTL scan function. The only problem is it's extremely slow, and that's because you have blocks in your model. So a way around it is to estimate best linear unbiased estimates. So using a sort of a mean, a weighted mean across your, in this case, your environments, by modeling the environments as a random term, a random effect in a mixed model, and then selecting your genotype effects as putting them as fixed effects. And then essentially what it just does is it gets you the means of your genotype scores, in this case for the number of prickles, across those environments, using allowing for some differences in those different block effects. Okay, so now we have a different phenotypic data frame for this trait. We're going to call it blue dot prickles on the stem. I'll give it a meaningful name. And if we run the analysis again, but this time we say the permutation test equals true. So usually that thing is false, but we just say, now give me a permutation test, and again, use multiple cores. So it first runs the QTL analysis, and it then estimates a significance threshold. Takes a little bit more time. And you can then plot the results of that analysis, and showing here quite a large effect QTL on linkage group 3, and also then two other QTLs, one on linkage group 2 and another on linkage group 6. Now they're very minor QTLs, they're just coming above the significance threshold, but it's quite clear that we have a major effect QTL on linkage group 3. Now, the importance of including these, especially large effect QTLs as cofactors, was pointed out well very early on in the literature, and Chris as well has talked about it, how you can do that. Now, you can either choose to do it in some sort of an automated fashion, or you can choose to do it in a more exploratory and a hands-on fashion. And like Polymapper, the polyqtlR philosophy is to allow the user as much control over those sorts of decisions in the analysis to us, and also to be able to do it quickly. So first of all, we need to define our peak, because our peak position, although it may not be where the QTL is, it's probably a good and probably the best first guess at where a major QTL lies. So we'll choose our peak position here. Funnily enough, there is no function in the package to find the QTL peak. So this is specially made for you. And of course, in an update to the package, I will include this. It's a very obvious function to have. And that just, for instance, finds the peak on chromosome 3 of our LOD, our QTL results here for the prickles on the stem. So what it says here is at position 25, we have such and such a LOD of 8.3, 8.4, and that the threshold was 5.5. So we found our peak on chromosome 3 at 25 centimorgans, so it's around this position, and we're going to now include that as a genetic cofactor. Now we use the same function, but now we introduce an extra argument in that function, and that's by adding a cofactor data frame to this. So it's a little bit, maybe a little bit counterintuitive, but this data frame, it doesn't really have to be named. It has 3 columns. The first is what is the linkage group group 3. The second is it a position or is it a marker? And that is important because in this instance, we're using a centimorgan position, which is 25 centimorgans. So it's a position cofactor. You could also use a marker as a cofactor, in which case you would provide a marker name here. Rather than a position, because often your genetic map has extremely long or precise or maybe overly precise positions for your markers, and wouldn't, it's probably better just to provide a name. there are some reasons also for that, and it's also because originally this package was designed with the output of tetraorigin in mind. So it's also, as I said, it's able to read in map poly objects. It's also designed to import IBD probabilities from tetraorigin, and in that instance, then yeah, you may have externally a file. So there are all these extra arguments available to read in and look at up folders and so on. But for now, I think the simplest for an exploratory analysis is to run that splining function and work with positions. You can always go back afterwards and run the analysis when you know you've got a QTL there using all your marker positions if you want to have a more precise indication of its position. But of course, estimating positions of QTLs at a resolution of less than 1 centimorgan in this kind of a population is probably a bit naive. So I would stick to this approach for now. Okay, so we've provided this as our cofactor data frame. So linkage group 3, it's a position and it's at 25 centimorgans. And we run the QTL analysis again. Now, as I said before, and Chris also pointed it out, significance thresholds are very fast to estimate with a permutation test, but only if your model is simple. So when you have start introducing things like genetic cofactors or environmental effects, so blocks, then the structure becomes more complicated and you can't use optimized, or at least I couldn't implement an optimized permutation test quite easily. So again, we have to go back to a sort of a simplified set of phenotypes like we did with those blues. How do we do that? This is, again, nothing very fancy about it. We just pull out the residuals of our model fitted at that cofactor position. So it's very simple. It's if one of the outputs of this function is to save the residuals if there is a cofactor included. So if we have a look at them here, these are just our genotype names, and then the residual phenotypic value once you have fitted effects as a fixed effect at that position that we provided. So this is the variation that's left over in your data when you include this position as a fixed effect in your model. So the phenotype looks a bit strange now. We've got -2 prickles and -4 prickles. Don't worry about that. It's mean centered. So don't. It doesn't matter that it's minuses and pluses. What is more important is that this is now corrected for that genetic that QTL, and we're going to rename it. We'll just call it residuals based on the cofactor and linkage group 3. Okay. So I've looked at that. So now the column names are geno and pheno. So they're what we're now telling it. So we're passing as our phenotype, we're passing now this residual thing as our phenotype data set as such. And now we're again running the function with the permutation test equals true, because as I said, your threshold can vary if you start introducing genetic cofactors. So you need to be able to re-estimate a threshold, and you need to be able to do it quickly. So this took about 10 seconds. So now we can plot the effect, plot the results there, and we can see here our peak on chromosome 3 has disappeared, and we've got a new peak that we hadn't seen before popping up in linkage group 4. But what's very interesting actually about this, something that was very unexpected, is that remember we had QTLs on linkage groups 2 and 6, but now both of those QTLs have disappeared. So usually the reason you introduce a genetic cofactor is to reduce the background variation in your data to detect more QTLs. So you'd imagine your significance at all your QTLs should go up, apart from the major QTL you include, because that is now gone, whereas the opposite has happened here. So it seems to suggest that there is some interaction going on, that once you account for the variation at linkage group 3, you're also somehow reducing the variation at linkage group 2 and 6. Now, it could be by chance that your map is terrible and you have markers from linkage group 3 mapped on linkage group 2 and 6. But if that was the case, you would have very, very strange linkage maps. They would be hundreds of centimorgans long. There would be massive amounts of gaps and so on, huge amount of stress. We saw yesterday that the maps we produced were very nice. So that's not going on. So it seems more likely that there is some interaction or epistatic interaction possibly going on between our major peak and those two minor peaks. Now you can overlay plots with each other, but if you do that in the wrong order, you can see, well, that wasn't very nice in terms of a visualization. And as well, you've got multiple thresholds here. I'm going to come back to this issue in a while and show another feature of this package, which allows you to do this much more nicely. But for now, let's go through the other cofactors. So we'll add the linkage group 2 peak as a cofactor. Again, now I'm just providing the position. I use the find peak function. It found the peak on linkage group 2 as 13 centimorgans. I've added that as a cofactor and I ran the analysis again. I save the residuals. So it's basically a lot of copy and paste work, unfortunately, but then you have a nice mapping pipeline in a single file. And again, this workshop file is available for you to use yourselves and to adjust and amend to your own particular trait data. Okay. Now, this is when we added linkage group 2 QTL as a cofactor. So we still have our peak on chromosome 3. And funnily enough, also the QTL on chromosome 6 is also lost here. So there seems also to be an interplay between 2 and 6. If we add our linkage group 6 peak as a cofactor, again, I'm just flying through these steps because it's the same thing again and again. And I'm running a permutation test. It takes a little bit of time. So here, our linkage group 6 has been included as the cofactor. And what we see here, again, we see that something on linkage group 4 now. A chromosome 3 peak is gone, which is, again, remarkable. This is our major QTL. But by adding a chromosome 6 peak as a cofactor, our chromosome 3 peak has now disappeared. And our chromosome 2 peak has somehow increased in significance. So there's quite an interesting interplay between these QTL, and this is something that you can pick apart quite nicely with this package because you have the ability to choose your cofactors by hand, add them to the model and test it. So I think, well, first of all, I think we should compare multiple analyses. We've run cofactors from our 3 original QTLs on 3, 2, and 6. I'm going to add the original data, so the original non-cofactor, so the naive model without any cofactors. I'll give them the colors that they had, and I'll use this function called plot linear QTL list. And the list means that I'm giving it a list of QTL results in this format. I'm giving them meaningful colors, and I'll add a little legend to the top corner there. So if we just take a moment to have a look at this, I'll zoom in a bit more detail. So this is our major QTL. And actually, the thing that popped out of this analysis, apart from the interactions between those QTL, is that we have another QTL on linkage group 4 that we didn't know about at the start in our green line here. And what you'll notice actually is if we add both the chromosome 2 peak or the chromosome 6 peak, we get an increase in significance, but the opposite happens. Sorry, I said that wrong. If we add the 3 peak or the 6 peak as our cofactors, we get an increase in significance. But if we added the linkage group 2 peak to that, we actually got a drop in significance. So there's some sort of dynamic interplay here, but it seems suggestive here that there is another QTL on linkage group 4, and it seems to also suggest that we should add 3 and 6 as cofactors in the same time. Okay, so you can do that because there's no limitation on the amount of cofactors you can add in a model. So let's build up our multi-QTL model by adding 3 and 6. Again, now we have a cofactor data frame is this. I'll just show it to you here. Yeah, on chromosome 3, that's our peak position on chromosome 6. So you can literally add as many as you want into a data frame and just pass that to this cofactor data frame argument. We save the residuals from that and rerun it again using the permutation test. So again, just to do it efficiently, time efficiently, because I can see I've got 20 minutes left, so I have to keep going. And now I'll plot that again, and we'll have a bit of a look at what that looks like. So here, oh yeah, I forgot to add the legend. That also helps me. Yeah. So here you see again the results of the analysis and again we get the most significance of all in the final analysis where we have 2 genetic cofactors added. So that seems to be the best background model, at least to describe the most variation or at least capture best what's going on chromosome 4. But in the end, almost all of those profiles would have given us an indication that there was something going on in chromosome 4. Now, there are other points of interest here. For instance, if you remember, on this chromosome, we had two peaks, and you might ask yourself, why is that? What's going on here? And that could, what you would do is that you would add your major peak as a cofactor and look at what happens with your secondary peak. This kind of thing happens. It's, I would say, attributable to noise in your IBD probabilities to some extent. So we're using here IBD probabilities that have been based on a bivalent model. Perhaps if we used the one based on a full multivalent model, we might have got less noise. So it could just be a reflection of that, or genotyping errors. You can go back, and Marcelo pointed out the advantage to increasing your error prior. So you can try, if you have noisy data, to maybe to reduce some of that noise. And I show it also in the vignette, an example of, in this case, is that the IBD probabilities we're working with today in rows, they're quite nice. But this is another example from another data set where, well, you can see you've got a huge amount of noise in the data, so massive amount of tension or genotyping errors, extremely long map lengths. So these kind of things can come out as well if you have poor quality data. And of course, you won't very much get a very clear association between these kind of probabilities and your phenotype. Okay, so there are some methods like increasing your error prior, or accounting for potential double reduction, which might clean up some of those profiles. But it is worthwhile, if you have multiple peaks on a single chromosome, just to see what happens when you add them as a cofactor. And there are instances that I've seen already, for instance, in potato, where you can find multiple QTL on the same chromosome at opposite ends, and that can be quite clearly shown using this cofactor approach. Finally, you can just use the results of that multiple QTL model results to estimate the position of your chromosome 4 peak. But actually, a single position for QTL is it's a little bit overambitious. A QTL peak doesn't really define a position of an underlying gene. It's much better to work with an interval, a support interval. And again, this function is hidden somewhere in the package, but it's not an explicit function in the package. It will be in a future update, but you have it here first. I'm calling it find support, just a simple function to pull out a support interval, in this case, a LOD2 support. So all those region within 2 LOD of the peak. And it provides the range of centimorgan positions there for you. Now we have a sort of a multi-QTL model here built up. We've got 4 different peaks identified and a position for them. There's a function in the package to look at the percentage variance explained, the PVE function. And that basically then just takes this tries to look at your, in the one hand, your 4 QTL model. So this is your full model, and then all permutations of submodels. So it's not quite like a backward elimination step, but it does allow you to look at how much variation is explained by your full model. So you can see here, we explain about 50% of the phenotypic variation using our 4 QTL model. And then you can also see, well, for instance, a 2 QTL model, which 2 QTLs together. So the one, it's QTL1 and QTL4. Now, QTL1 has nothing to do with linkage group 1. It's just how we define them in this. So that's the big one. And the one that we found that seems to be independent to the big one, so on linkage group 4. So those two together are quite a good parsimonious 2 QTL model. Now, what you decide, how many QTLs should you have in your final model, that's a little bit, I would say, up to you. Of course, in the end, there'll be no more significant QTL to find. And in this case, I think we're happy enough with a 4 QTL model, but you can, again, look at those submodels and see how much variance is explained by those. Now, the last topic that I'm going to cover really for today is the topic of exploring the QTL peak. So finding out what's the most likely segregation type at the QTL. And that really, well, it can be done either using the Bayesian information criterion. And again, we're following the lead here from Christine Hackett over in Dundee. This was something that the two of us were looking at when we were looking at that study with double reduction in it. We found if you include double reduction in your model, you actually have many more possible QTL models to fit, and the fitting, the predictive ability of this BIC procedure tends to go down. So you're not as accurate in your prediction of your QTL type, because I suppose your prediction space is much bigger, and you have the potential to fit an incorrect model. Now, what does this visualization show you? Well, it shows you all the different QTL models that you're testing. There, we can have a look at it. There's a sort of a default set of models that's provided with the package, but you can provide whatever one you want. We have gone a step further to look at multi-QTL models, but that's not a default setting in the package at the moment. But that is something that I can add if people are interested in testing multi-allelic QTL model, I meant. So having multiple, not just a single allele, but having multiple alleles with different effects. That's also possible. But what's probably a bit more intuitive is this function which visualizes the QTL effect. So we're looking here at the peak on chromosome 3. So that's the major QTL. It's spread out a little bit here, so it doesn't look as sharp. But here again, you see the effect size in terms of number of prickles for each of the different parental homologs. Now, it differs a little bit from what Chris showed, and that's because the population, the genotypes are a little bit different, and also the ordering of the homologs is, of course, random each time you estimate the phasing. So in this case, yeah, the numbering, but there is a single homolog, it seems, from the first parent that contributes an extra 2 prickles. And I remember thinking to myself, from a breeding perspective, would a rose with 2 fewer thorns be assailable? But in terms of the range of values that we have for this trait, 2 fewer prickles is actually quite a big effect size in terms of the scale of the number. So indeed, 2 fewer thorns might well be a selling point. But in any case, that's not maybe the point here. But if you did want to develop a marker for that trait, you can then go back to your phase map and find markers around that region that are tagging or at the same segregation type as that. So that would be a potential application step then towards marker. It can be interesting as well to visualize your haplotypes and connect them with your phenotypes. So previously we were displaying our haplotypes, so these probabilities, displaying them by, I think, individual, if I'm not mistaken. I would have to look at, no, okay. I would have to look at the help menu for that function, but in this case, I'm displaying them by phenotype. And that means, so I'm sorting the individuals in the population by the trait. So I'm getting the most extreme individuals for the trait, and then I'm lining them up and I'm showing as the highlighting the region around the QTL. So that's the support QTL support region around the QTL. So I have here basically the QTL peak in red, and then I have the individuals. And remember from our previous plots here, we knew there was something going on with homolog 2 around that. So what we would do here is look in those individuals, and of course the first individual is an exception. That individual, although it has a lot of prickles, it doesn't carry homolog 2, so that's not good. But for the rest of them, they all carry homolog 2. So you can try to piece together those individuals, pull out those individuals' extreme values, and see, do they fit with the predicted model, QTL model, that you found at that peak. So that can be quite useful, I think. Now, there are extra material. I think I'm maybe going to skip over them. I'll just tell you what they can do. The first question I think earlier was, what if you don't have a map? Maybe if you don't have a physical map or even a genetic map, well, you can do a single marker approach. If you have a map, then you can plot them. I suppose I can swap over to the vignette where this has already been done for me. Yeah, so this is, there is actually a little bug in the visualization function at the moment. If there are unmapped markers in your data set, which often there are, and I fixed that, but I haven't pushed it to CRAN yet. So you have to do a little correction on it. I might point out the correction that you need to do to fix that. But anyway, this shows you the overlay between the sort of a single marker approach in blue dots and then your IBD based approach. You can see quite a bit of consistency in terms of the results. On linkage group 6, though, for instance, you find a QTL peak, but that falls well below the significance threshold using the single marker. So there are advantages to using these IBD probabilities, which can even be found in a data set like this. Oh yeah, I should still be on my vignette. And then, Okay, extra material, meiosis recombinations, all these kind of things. So if you're interested more in the biology of meiosis and the what went on in your parental meiosis, there's a huge amount of information contained in those IBD probabilities, and the some functions are provided here, for instance, to generate a meiosis report. So um, looking for deviations from the model of random pairing between your homologs. So again, these sorts of things have also been shown, I think, yesterday by Marcelo, um, in terms of looking for preferential pairing. So this function as well does something similar. It uses the IBD probabilities at the output of the hidden Markov model to actually look for extra interesting things. The last thing that I do is talk about recombinations, a recombination landscape. I think it's a very nice point to end on, is that you can go back, for instance, here you can count the number of recombination events per individual, and you can see here there are some individuals that had a huge amount of crossovers, a huge amount of recombinations in those profiles. And that's also in other mapping software usually provided, and it's nice to have it also available in Polyploids so that you can then go back and that individual probably has something wrong with the DNA, and maybe you go back and even recreate your maps without that individual. They might get a little bit shorter at least, and maybe a little bit more accurate. Yeah, and you can overlay those predicted recombination breakpoints. You can overlay them with your IBD, your visualization there as well, so that you can see that everything is working out nicely. So indeed, here is that individual. Just to end now, Dave, we can switch to questions. That's in that individual which had a double reduction, but here there was all these extra recombinations predicted because double reduction wasn't accounted for. Yeah, and that's it. So it's available on CRAN. If there are questions, I'm happy to take them now.
This presentation has been sponsored by the Tools for Genomics Assisted Breeding and Polyploids Research Project. For more information, you can email us at toolsforpolyploids@tamu.edu or visit our website at www.polyploids.org. This project is funded by NIFA within the USDA Specialty Crop Research Initiative funding program.